In this module, we will be talking about molecular biology lab equipment and their proper use. If we were in the lab in a face-to-face setting, we would have gone around the room and looked at all the equipment present in your molecular biology lab classroom. However, since we don't have that luxury, we will have to do with the pictures and their text. So, let's start with personal protective equipment, or PPE. These are things that you bring into the lab yourself many times in order to protect yourself from the hazardous materials that are going to be present in the lab. This includes safety glasses and goggles, which must be worn during all labs, regardless of whether you're working with biohazardous materials or not. They protect our eyes from any chemical splashes, a fire, or any biohazardous material that may spill. And these are extremely important. Another thing that you should have for most of the labs, if not for all labs, are gloves. You should definitely be wearing gloves anytime you're working with biohazardous material or chemicals that can harm you, but you should also be making it a good practice to work with all labs using gloves. When we are wearing gloves, we have to be aware of a few things though. The gloves that we get are not sterile on their own. We can disinfect them and sterilize them by using hand sanitizer once we have worn them, and then periodically during the lab. In addition, if we take our gloves and touch our face or hair, then they no longer remain clean either, and they will be the source of contamination in our system. Therefore, you should be mindful of that and make sure that you do not do so. If you do accidentally touch your hair or face, you should disinfect them with hand sanitizer again. If you need to leave the lab for a break, you should leave the gloves behind if you haven't finished the procedure yet. You should never touch the knobs of doors with gloved hands, because you don't know what chemicals or substances may be on them. Your hands are protected, but now you've transferred that stuff onto the knob with your gloves. Finally, once you have finished the lab, you should dispose the gloves in the lab before you leave. Those gloves will usually be disposed in either normal trash or in a biohazardous bin, depending upon what it is that you were doing that week. The next equipment we'll talk about, this is more a consumable, is a serological pipette. The serological pipettes essentially are mini cylinders. They allow us to measure relatively small volumes, anywhere from 1 to 25 ml during experimentation. They come in various sizes based on what it is that we need. The smallest one is usually 1 ml. Typically, you don't see them in volumes larger than 25 ml, since you can easily use a graduated cylinder for those purposes. We use pumps that can be either manual or electronic to fill up the liquid inside these pipettes. When we are working with cell culture under aseptic conditions, we use special serological pipettes that are individually wrapped and are sterile to maintain our aseptic technique. When using these, you should always be mindful of where the tip is. The tip should always be facing away from you. You should work with them, keeping them vertical at all times. When you are using the pumps, you should choose the pump. If you're using the manual pump, you need to use the pump that is appropriate for the pipette size that you're using. We will learn more about this in the pipetting skills. Mostly, when we are working in molecular biology labs, even the 1 to 25 ml volume is a large volume. Typically, since cells are so small, the materials that we get from them are small as well. So we need to have an accurate way to measure super tiny volumes. We do that by using micropipettes. Here is an image of a micropipette. Micropipettes are essentially super accurate calibrated pumps for pipette tips that fill up with liquid that we need to use. The liquids that can be picked up with micropipettes are anywhere from 0.2 ml, 0.2 microliter, all the way up to 1 ml or 1,000 microliter. So these are for small amounts of liquid, very small amounts of liquid. There are different types and different levels of accuracy depending on what company you get or what accuracy you get. But most of them usually go to the nearest microliter. These micropipettes are used in conjunction with microtips. The micropipette tips are special tips that are made of plastic that can be autoclaved so that we can make them sterile if needed. However, you can also find them in sterile DNase and protease-free, RNase-free systems available from the market. Most of the time, however, we just use disposable autoclavable tips, and they are the ones that we are going to be using to pick up small volumes of liquid with the micropipettes. They are disposed of after each use, unless you're taking the same liquid again and again, in which case, as long as you keep your technique good, you can reuse them, going from the lowest to the highest concentration, or going from one well to the other, as long as there's no chance of contamination. To make things sterile and to decontaminate waste, we use autoclaves. The autoclaves removes microorganisms, which are usually virus, bacteria, fungi, or mold, as well as their spores, using a high pressure, high temperature steam sterilization system. It can be used both for solid as well as liquid substances. It is used to sterilize contaminated material like your biohazardous waste, but it is also used to sterilize glassware, plastics, and pipettes, so we can use them in experimentation and techniques. We can use it to sterilize liquids and solutions, so they can be used in experimentation and can be kept for an extended period of time. Another instrument that is extremely important in molecular biology lab is a spectrometer or a spectrophotometer. It produces a light of whatever wavelength that you need, and which passes through your sample, and then it is measured at the end with how much intensity is left behind. It reports the value as the amount of light that was absorbed, or the absorbance. The higher the absorbance, the more substance is there. We use this to measure growth of microorganisms like bacteria and yeast, or any other cells that may be grown in suspension. We also use these to find concentration of proteins or nucleic acids in our extracted samples. We can use it to detect contaminants, and we can use it to run activity assays, which are colorimetric or fluorimetric. To prepare our samples, many times we need to separate several different components in the cell or in our solution. An example of that would be needing to pellet just the nuclei so we can look at nuclear protein or the cytoplasmic protein. Or we may be interested in looking at biological fluids and we want to look at the cell-free serum versus the cell pellet. In that case, we take advantage of centrifuges. A centrifuge is an apparatus that rotates at super high speed and separates substances based on their densities. So the heavier things will be at the bottom, and so on and so forth. We use it to remove insoluble matter from extracted proteins or nucleic acids. We also use it to separate subcellular components like nuclei, mitochondria, or we can use it to remove cell elements from blood so that we can get cell-free serum or plasma. In addition, certain methods use the solubility of a substance to reduce the solubility of the substance to get our DNA, RNA, or proteins out. In that case, we pellet our DNA, RNA, or protein or lipids so that we can look at them in an isolated fashion. To use the centrifuge, we need special tubes that will not burst or crack under the high G forces that are induced by the centrifugal forces in the microcentrifuge. These are specifically designed, usually tapered, and they usually have this little, conical shape. Even when they are flat and so that they can stand, there is a conical shape underneath, separate from it, to make sure that it does not crack. These specially engineered test tubes are what we use in the centrifuge anytime we need to use it. We have to make sure that we're using the centrifuge tube of the appropriate size in the appropriate centrifuge though. Otherwise, they still crack and burst and lose our sample. For microcentrifuges, the tubes can be anywhere from 0.2 ml, which would be 200 microliters, all up till 2 ml. And for macrocentrifuges, you can have tubes that are 5 ml, 10 ml, 15 or 50 ml. All of those tubes can be used in those systems. Other times we are doing experiments where we don't need to separate our sample, but rather we want to create a homogeneous solution or suspension. If that is our goal, we use a vortex. A vortex basically stirs our sample at a controlled speed so that it can mix it and make it into a homogeneous solution or homogeneous suspension. We use it to mix cells in suspensions, to mix reagents of an assay so that the entire system is homogeneous and acts the same way. And that helps us in getting reproducible results. Another thing that can help us with accuracy and reproducibility is a water bath. When we need to heat our sample at a constant temperature, instead of using a dry heating block or a dry incubator, it is better to use water to heat or maintain the constant temperature. We use this water bath where it essentially has an insulated outer shell and a tank inside that you can set to whatever temperature you need. We use these to heat or thaw fragile samples, such as mammalian cells, when we first take them out of frozen stock. We also use it to maintain a homogeneous temperature control when we are working with nucleic acids or proteins during extraction procedures or during activity assays. Now, sometimes we can just incubate our samples on our benches or in the water bath at the temperature we need. Other times, we need to incubate our samples in a way that they are constantly moving. Now, if you have an experiment which requires you to shake your sample or to mix it or agitate it for 45 minutes as it's incubating with a particular antibody or a chemical reaction, that would be really cumbersome to do with a lot of tubes. So, to take our hands out of the equation, we use shakers. The shakers can be found in various forms. Some of them are flat and they rock back and forth, or they can go kind of move around, and those are called belly shakers. Or they can just shake in one spot and agitate our samples and aerate it in an insulated environment where you can also set temperature control. All of these shakers work to mix or agitate our solutions or our samples before we place them into a spectrophotometer or a centrifuge for the next step. We use them to incubate antibodies, chemical reactions, and such things during experimentation which need to be constantly mixed to work appropriately, and this allows us to get reproducible results. We can also use them, as we are using it down here, to aerate our media while growing bacterial or yeast cell cultures. Now, as we are working in a molecular and cellular biology lab, we need to look at molecular things, which involve looking at a lot of cells. Now, cells are small and they are not visible, majority of them at least, are not visible with naked eye. So we use the help of microscopes, both compounds as well as inverted microscopes, to visualize our cells. For typical slides that we have or that we prepare, we can use a simple compound light microscope, which uses a light source at the base of the microscope and a nose piece with objective lenses on the top of the stage to magnify our samples anywhere from 40 to 1000x. On the other hand, if we are growing cells in culture where the cells are attached to the bottom surface and then have a media on top, this system, the simple compound light microscope, will not give us a lot of good resolution and will make our images blurry, since the nose piece with the objective lens is on the top, and it would be looking through a liquid media before looking at the cells. In that case, we use an inverted microscope, where the nose piece is at the bottom and the light source is at the top. And in this way, we are able to visualize the cells that are grown in culture in a much better resolution. And they can, again, magnify the objects anywhere from 40 to 1000x. To grow the cells in culture, we use the help of water-jacketed CO2 incubator. Just like a water bath maintains a more constant temperature control, a water-jacketed CO2 incubator does the same. The water coils that are in between the inner and the outer chamber all around the incubator maintain a more homeostatic environment inside the incubator. Many of them also have humidity and CO2 control. The CO2 tank maintains the same CO2 oxygen concentration like it would have inside our body. We use this to culture adherent cells under specific conditions, either mimicking our normal physiological conditions or diseased ones under some circumstances. For example, we could mimic the emphysema or low oxygen tension by removing the oxygen from the system to a lower concentration than normal, or creating hypoxia by cobalt chloride. To grow the cells, we need a vessel to put them in. We can't, in our body, they have our bodies to work with. However, here we use either glass or plastic dishes or flasks. Sometimes we use cover slips on slides. Any one of these things that we will use, they will have a coating at the bottom that will mimic the extracellular matrix, so the cells can adhere to them and grow and replicate. We use these to culture mammalian cells in vitro. And we can also use some of these to hold culture medium and agar so that we can grow bacteria and viruses. Once we've grown our cells and extracted the molecular material that we need, either DNA, RNA, protein, usually we next want to visualize it in some way, shape, or form. One of the ways that we can separate the proteins and the DNA in a given mixture and then look at it is through gel electrophoresis. When we have a DNA sample, we usually use a gel made of agarose. And when we have a protein sample, we usually use gels made of polyacrylamide instead. The DNA gels are usually horizontal, and the protein gels are usually vertical. However, that's not a steady rule. There are DNA sequencing gels that do run vertically. Similarly, there are protein gels that are separating protein by their isoelectric points that run horizontally. However, in either case, the idea behind this is that the samples will move and separate based on either charge or by size. In this case, we are separating the DNA or protein by their size alone. DNA is negatively charged naturally because of the phosphates in the backbone. So in this case, when we run the power source, the DNA will automatically move from the negative to the positive side. However, proteins can have different charges based on the R groups and the overall charge on the protein. In those cases, we can run those proteins in their native conditions without changing those charges, or we can add a reagent such as STS into our extraction procedure so that all the proteins become negatively charged and separate solely based upon their size. In both cases, the proteins or DNA that are smaller, the fragments that are smaller will move further, and the fragments or the proteins that are larger will be closer to the top. So we can look at that along with a molecular weight marker and know exactly the size of the sample that we are interested in and that we are looking for. So these are some of the lab equipment that we are going to familiarize ourselves and that we are going to learn more about over the course of the semester. In addition to these, we are also going to be using biosafety cabinets in our virtual simulations, as well as our fume hoods. Those are things that we've talked about last week. Make sure before you do your simulations or work through your simulations for this week that you do run through this PowerPoint as well as any other material that I've posted in the module so that you're ready to take the quiz for the week and do the assignments for the week.